Hi everyone, welcome to Bio3050 Advanced Cell Biology. I want to use this time to go over a little bit about the course expectations, the course format, and guide you a little bit through Canvas. This way we don't have to spend a lot of time the first day really just going over the syllabus and through a lot of things that you can read on your own, and you guys can probably figure out by now since you're in upper division. And that way we have a little bit more time for some things that are more fun when it comes to the first day in class. So here we obviously have the home page of the Canvas course, and this is where you can find out a little bit about me. This is also where you can find the course syllabus, and probably the most important part later on in the semester is this modules, which I'll talk about a little bit more. But let's jump to the course syllabus. Here you can find the syllabus and also the general course schedule. The course schedule is also on the syllabus, and also the lab syllabus, and I'll talk about each of these in turn. So if we jump to the syllabus, this just has the standard information. So we're meeting in Seder Hall 120. Hopefully by now you know when the class meets, Monday, Wednesday, Friday, 11 to 11:55. And then the lab is Thursday afternoon, also in Seder 120. Pretty much here's my office hours, but honestly, you guys, if you know me by now, you know that all you have to do is email me or stop to ask me at the end of class or whatever. Well, let's find a time to get together and I'll make myself available. So don't worry just about the office hours. Even with the office hours, I tend to be pretty busy, so you might want to make an email appointment just so that you know I'll be there. But I really do want to help you, so I will make myself available as much as I need to discuss whatever it is you want to discuss. So there's the PLNU mission, the course description. These learning outcomes are important because this is what you should be able to do by the end of the course, what we're going to learn. Evaluation and grading and so forth. This is pretty standard. And here you can see the different grade distribution. So you can see the lab is a big part of it. We'll talk about the lab a little bit more. But a lot of this, as you're going to learn, is it's a discussion format called Jigsaw. All right, and so this is a little bit of a different format than you're probably used to. And so within this, there's a lot of participation and group teaching and things like that that are going to be a big part of this. So really just being a good participant and a good peer and teammate and classmate is going to weigh a fair amount on your grade as well. The textbook is the big blue Alberts. I know this is huge and we'll only be using a part of it, but this is actually a really important textbook that's used quite often in both graduate school and medical school. So if you go on to either of those, you will definitely probably see this again. And even if you don't, it's a really good resource, one that I continue to use just in my own research and so forth. So hopefully you find it is a helpful and informative book. All right, so this format of the class is a little bit different than most of the ones you've probably had before that you've been used to. So this is taught in a jigsaw format. This is so that you're actually an active part of your own learning. Basically, there are 6 units in the course. 2 of them, units 1 and 4, are going to be a little bit more conventional where I'm doing most of the lecturing. But even when I talk about lecture, we're still meeting in groups. We're still working together on applying concepts in higher order learning and so forth. So it's not like I'm just throwing out information at you. But those are a little bit more conventional. The other 4 units are the jigsaw format where basically you guys are all divided into 4 different groups. And group 1 will have 1 portion of the material. Group 2 will be in charge of another portion. Group 3 another portion. In one of the groups, each unit will be in charge of learning about and presenting a primary literature article. And there are worksheets along the way to help guide you through your portion of the reading and what I really think is important, what I want you to be able to know. And then you're going to be in charge of teaching each other kind of those portions, and then we're going to use that information together as a class to really solidify some of that knowledge and apply that knowledge to higher order thinking and so forth. So that allows us to really work together in class to take that knowledge and really apply it to the next level learning as an upper division course. So I'll talk a little bit more about this, but this is one of the reasons I did want to introduce this course in this course orientation video, because that part's a little bit different. All right, so as we go through here, you can see this is the general timetable within each of those 4 units that are the jigsaw units. So the first day we're going to have an introduction, and this is where I'm basically going to go over some basic information about the unit, giving you guys a lot of information that you might need to really understand your own portions and then be ready to teach. And then there's going to be a worksheet that is going to guide you through your portion of the reading that you have to answer. And you're going to answer that prior to the same group discussion. So the next day in class, for the unit, you're going to meet in the same group. So this is in people that were also in group 1 if you're in group 1, or people that are also in group 2 if you're in group 2. And so everyone's read the same material, answered the same worksheet questions, and this gives you a chance in class to really help each other understand some of the concepts that maybe you struggled with a little bit. And I'll be walking around throughout this class, answering questions and so forth. But this also gives you a chance to practice teaching to each other some of those key figures and some of the things that you're then going to teach to your mixed group on the mixed group day. So starts with introduction in class, and then the next class day is the same group discussions. And then within that, you're also going to start to work on your 2-page summary. So the whole purpose of the 2-page summary is to take that usually about 20 pages of your reading and really figure out what the take-home messages are, what are the key points, and then summarize all of that into 2 pages that you're going to be able to give to your mixed groups. So that way your mixed groups kind of have that summary of your portion, and you're going to get those 2-page summaries from the other people in your groups. Then the next 2 days of class are mixed group work. So this is where you're actually meeting in those mixed groups, and each group will have about a half hour to kind of teach their portion to the other groups. And again, I'm walking around helping clarify misconceptions and helping kind of apply or put this into perspective with the class and so forth. But really you're just teaching each other and learning from each other during this time. And then that leads to what I call concept clarification. And there's usually about 4 days worth of class for this. And that concept clarification is where we're now applying all those things that you've learned together. And I'm going to lecture some, basically helping clarify some of the misconceptions and the things that I noticed people were struggling with along the way. The reason for doing the mixed group or me walking around and kind of helping with that is I also will know the parts that you guys understood really well. So we don't have to spend time together in class going over those things that you really got. We can focus on the parts that were a little bit more difficult, and we can actually focus on using that information at the next level and really applying it in some mixed group problems in some biomedical types of applications and things like that. All right, so homework assignments, the big one probably is the worksheet. This is what's guiding you through the reading. I'll show you that on Canvas. Then there's the 2-page summaries that you have for your content that you're going to provide to your mixed groups on the mixed group teaching days. And then there's also mixed group problems. These are the kind of part, we're going to be doing a lot of these in concept clarification together, but there will be some that you have to do on your own outside class, but you're doing all of these in your mixed group of students. And each mixed group will have one person that was in group 1, one in group 2, one from group 3, and one from group 4. Excuse me, and you're merging all that material together. I've also started to make, or have made these crossword puzzles. So one of the things with cell biology is there's a lot of information. This is one of the reasons that the jigsaw works well, is because we can kind of get some of that base information down and then apply it together and use the time, a lot of the time together, really to take it to that next level. But that also means there's a lot of terms. So I created these crossword puzzles as a hopefully semi-fun way to really familiarize yourself with some of the key terms. And so these crossword puzzles even have pages where you're going to look for those terms, but you're just basically finishing the crossword puzzle as part of a way to learn those terms. Over the years, I've decided that people don't always want to do all of them, so I required everyone to complete the first unit crossword because that's a baseline background information that you really need for the whole semester. And then you only have to do 3 of the last 5 units. If you find them useful, you can do all 5, but basically you have to pick 3. And this gives you a little bit of flexibility if there's a time when you're just really swamped with other projects and other classes, etc. So trying to give a little flexibility there. I talked about the group participation. Attendance in class is really, really important because you guys are going to be teaching each other, and preparation to teach each other and all that. That's very important. So there's opportunities for you guys to evaluate each other and really just hold each other accountable. If everyone does their job well, these are some free points, but it really makes sure that you're ready. And you want to be accountable to your peers anyway, because you want them to do well in teaching you, and you want to do well in teaching them. Midterms, final exams. As I said, one group for each unit has a primary literature presentation. That's going to rotate. So if group 4 has the primary literature presentation for unit 2, it might be group 3 for unit 3, and group 2 for unit 5, and group 1 for unit 6. So all of the kind of different parts of the reading and the different which group has the primary literature presentation, that all rotates around to make it even throughout those 4 units. I'm going to give a lot of feedback, so on the worksheets, for example, if you guys turn them in early enough, I will provide feedback, and that feedback is there for you when you get into the same groups to really use to make sure that you are clear on everything there. So make sure you're looking at my feedback in Canvas and take advantage of that. I'll talk a little bit more about the laboratory work in a bit. All right, and the rest of this is just kind of standard syllabus stuff. So if we head back to Canvas, we go to modules. This is where you're going to spend most of your time in the course. And you can see at the first module there's important course information. Oops. All right, so that's where the course orientation was. Learning objectives are really important. This is basically the study guide, or the learning objectives, the things I think you should learn from each of the units. So these can be used as a study guide when you're studying for the exam, but I also highly recommend you taking a look at them before the unit starts and paying attention to them during the unit so that you really know kind of what you should be learning as you go. That kind of gives you some expectations and some things that you know to look for. Another thing you'll find is some more important information here. The assigned groups for unit work. This is going to tell you what group you're in. So whether you're in group 1, group 2, group 3, group 4, so that's really important when we get to the units so that you know which worksheets to do and so forth. So make sure you pay attention to that. I've also got some examples of 2-page summaries so that you kind of can see a template or an example of that when you start writing your own. This is information on the primary literature and rubrics and things like that. One more thing to take a look at is the Henrietta Lacks book discussion. So we will be reading Henrietta Lacks. And Henrietta Lacks basically divides itself into 3 parts. And so you'll see this on the schedule that I'll show in a second. But this is where you can kind of participate in the discussion boards and things like that. And then some more important information here, including how the change of speed of an ed puzzle, if you want to speed it up a little bit and so forth. I wouldn't speed it up too fast, but you can speed it up a little bit as long as you're still getting the information and taking notes and finishing the questions along the way. All right, let me jump to the last part of the syllabus here. All right, so if we go to this back to the syllabus, here's the course schedule. And let's jump to Unit 2, for example. So in Unit 2 here, you'll see that we have an introduction, like I talked about, that first class. Then in the group work, this is where you're going to be in the same groups. And so your unit worksheet is due by then so that you are able to participate in your same group and help each other with misconceptions and clarifying things that you maybe didn't fully understand. And again, if you turn in this worksheet early enough, you'll have some feedback with which to work in that same group. And then you'll have your 2-page summary due by the next day, because that's when you're going to start meeting in your mixed groups for 2 days. So introduction, 1 day in same groups, and then 2 days in the mixed groups. And then there's about 4 days of concept clarification. And this is where we're really applying all of this together in class, and we're doing some of the mixed group problems and so forth. And as of COVID a couple of years ago, some of these became a little bit asynchronous, and it actually works pretty well. And so I've taken the topics that I know people struggle with and work pretty well in asynchronous, and we'll continue to do that as long as it works. So this will give you some flexibility where some days you can actually learn the material via a video and an ed puzzle activity and so forth. So there's about, I don't know, less than probably like 8 of those scattered throughout the semester, just to give us some breaks where there's some opportune times for that. But you do need to do those ed puzzles, work with that asynchronous activity, and treat it as if it's a class, because all that information is still just as important as if we were meeting together. You can see that the exams are going to be in the lab, and that's kind of what the exams are on. And then at the end of a unit, there's some mixed group problems. In the group evaluation, you evaluate each other, and then we start the next unit. All right, so here's where we have the Henrietta Lacks. So their Henrietta Lacks, the book, is divided into 3 parts. And so you can see the 3 parts there. For each of these, there is a discussion board that is due on the Monday. And then you have to respond to your small group peers by Wednesday, and then we'll actually talk about the book in class. So this is a wonderful book. It gives us a lot of information on some inequities in science. So a lot of discussions, bioethical discussions to have with that. And it also has a lot of good historical background on cell culture and how that came to be, because in lab we're going to be doing a lot of tissue culture, and that tissue culture is one of the key aspects of cell biology experimentation that you'll not only learn, but we'll learn a little bit about the history of it. All right, so that's how you can kind of find, use the course schedule to find out the topics, the dates, the textbook parts that we're talking about, and then what's due kind of in each of these portions along the way here. But that information is also found in Canvas. So again, if you go to the Henrietta Lacks book discussions, this takes you to each of the discussions. There's some information here, but the real information is going to be in the discussion itself. And so you have the actual prompts, right? So you, in this case, you have to respond to 5 of the 7 prompts. So basically on your discussion board, you're going to answer 5 of these 7 ones by that Monday. And then you're going to respond to the one of the prompts for each person, all right? And you're really going to challenge them to think, and just really, this is a great way to engage in a bioethical discussion and so forth. So those responses are due by Wednesday, and then we're going to discuss as a class in full in lab, usually during some incubation times, etc. All right. So if we go back to modules here, the other thing I just want to point out is that 2 things. You can find information as we get down to kind of the meat of this. You can find all the information for a unit in kind of this unit overview. So each unit has a unit overview. And within that, there's some information on the unit itself. So you can read about that, and it talks about what you should be learning, do the overall learning outcomes, things like that. This is where you can also find slides. So I'm going to try and provide my concept clarification slides ahead of time, so you can use those for notes and don't have to write down everything as long as you go. I highly recommend that you kind of print these out, or you use it on your tablet so you can write your own notes on the slide, and you can put the information that you need to really understand it later. But these can be printed out 6 slides per page, and while my actual slides may vary on it a little bit, I will try to keep these as up to date as possible so that you have that information right there with you, and you can just write the key take-home messages and the big ideas and so forth, instead of trying to quickly jot down everything as we go. All right, so if we go back, there's the unit overviews. Really importantly, we also have, all right, so if we, it's jumping around on me, but there's the week 2. Learning objectives again, lecture slides, interesting videos. These are some tools and resources for you. The group reading assignments, this is where you're really going to need to pay attention. So if you know what group you're in already, it'll tell you what portions of the chapters you need to read. So in Unit 2, we're reading chapters 10 and chapters 11. And then in this case, Group 4 has the primary literature article. So that's the assignments that you're, that's the portions that you're assigned to read. And then we have the worksheets. So this will also tell you what you're expected to read, but this gives you the worksheet that you're going to, with the questions you're going to answer along the way. So these are guided questions, really kind of to help you understand what I think is important from that reading, and that you're going to then kind of focus on with teaching. And it also has some key figures that you should be ready to teach when you get to the mixed group problem. So that's one of the things you guys can practice teaching each other in that same group day. All right, so this is kind of all the worksheet information that you need along the way there. And then if we jump back still in the Unit 2 overview, the 2-page summary, crossword, mixed group problems, those are kind of the assignments throughout it. And then you'll see that every week basically just has a format that tells you what's going on that week. So you have the things that are due before each day, like this worksheet is due by that Monday because that's when we're meeting in same groups. Remember, the worksheet's due before same groups. Then the 2-page summary is due before the mixed group and peer teaching. And then there's information on what we're doing that week in lab and some other things that are coming up soon, just reminders of homework that might be due pretty soon. And so you can see we have that for each week, so you really shouldn't have any question of what's due when between the syllabus, the schedule, and these weekly modules. It should be hopefully pretty straightforward for you. All right, last thing I want to talk about is the lab. So Thursday labs, basically we are going to have 3 projects. One of them is a really extensive project and then 2 smaller projects. The extensive project is learning how to grow cells in culture. It's called tissue culture. And then we're also going to use those. So for the course of about the first 6-7 weeks, you're going to be growing cells in culture. You're going to be maintaining them, caring for them, splitting them, These are all things that we will learn on the very first day of lab. And then you're also going to be preparing them for various experimental methods that we use often in cell biology. So you're going to do immunocytochemistry, where you're staining them cells. We're going to learn confocal microscopy. You're going to be doing a viability assay, which basically is testing the effects of chemotherapy on these cells. We're also going to stimulate cells with some cytokines and use some inhibitors, and we're going to figure out what happens to the gene expression and protein expression using Western blots and so forth. So we're going to be doing a lot of things with these cells that you're growing. You're also going to do some cryosectioning. You're going to create your own tissue slides from mouse brain, and then we're going to do immunohistochemistry of that. So we're going to stain that and use confocal to watch it, to image that. And then the last couple of days, we're going to be growing stem cells in culture and differentiating them and so forth. So a lot of fun stuff to do. So all of these things are handouts. There is no lab notebook. I've kind of created these handouts for all of these that you'll need to read ahead of time in lab. And the big project is maintaining an e-notebook. So a big part of lab is basically keeping track of what you did, the results, and the context of all of these things. So we're going to be maintaining an e-notebook for all of the tissue culture in a cryosectioning portion, and then we'll just kind of have a little bit of a fun lab at the very end. But there's a lot of information here on the e-notebook, so make sure you take a look through the lab syllabus, and it even tells you pretty much exactly what I expect to see in the e-notebook for all the different little portions and so forth there. The one other thing that you need to know is these handouts do need to be read ahead of time. So in the weekly modules, you can actually see kind of what we're doing in lab. There's a lot of information there, and then there's handouts and things like that that need to be read prior to lab that day. If you wanted to find this in a more centralized location, there is a unit at the beginning here with the general information that tells us the lab information and protocol files. So these are the kind of different protocols, and then some other notes and things that can be useful and so forth. And the one thing that you absolutely need to do, in lieu of a quiz to kind of hold you accountable for reading this, what I want to see is your e-notebooks. I want to see the purpose and the introductory paragraph that kind of introduces what you're doing and why and what we mean to do, and then just a general information on the methods section that shows that you understand the methods and what that entails and so forth. So if you take a look on here, you can actually see that pre-lab work and what I expect to see prior to the lab, and then we'll continue to maintain those e-notebooks basically with what we do in lab and what we accomplish and so forth. So I'll talk a little bit more about the expectations there at the first day in lab as well. So that's it for an overall introduction to the course. Make sure you take a few minutes just to play around with Canvas. I hope I've organized it in a way that's really easy to navigate and find what you need. And also just take a little look through the two syllabi, the course syllabus and the lab syllabus, and read those beforehand, and then bring questions to class that first week, and then I'll be happy to go over anything that you have questions about. All right. I really look forward to it. This class is a really fun class to teach. Students really seem to get a lot out of it, and I've had a lot of students write me back out in graduate school and medical school and say the format of this class has really helped them to remember things and to really apply these things to long-term memory, and it's helped them for both work, graduate school, and medical school. So hopefully the same is true for you as well. Looking forward to it. Thank you. Bye.